Hello, my Form 5s. Welcome back to our weekly class for July week 2. Okay, today we are going to continue on our chapter 5 inheritance. Okay, so before I begin, let me just tell you that this part of this chapter is not very common in exam. Okay, so I wouldn't recommend you to focus too much on this. Okay, the last week stuff we have learned is still the more important ones. Okay, so the crossing diagram, remember, okay, Mendel's first and second law is still the important ones. All right, so let's begin. Last week we talked a little bit about this, just a little. Okay, basically we talked about how DNA forms. Okay, so let's begin. You have a cell. Okay, you have cells, millions of it, trillions of it. Okay, in a cell you have nucleus. Every nucleus has chromosome. For us, we have 46. Okay, 23 for mama, 23 for papa. And then on the chromosome, you have sections. Okay, so you have sections. Okay, I told you about this. Let's say one section for height, one section for skin color. So that is what we call gene. Okay, gene, this is what we talk the most about. Okay, so a gene is made up of tiny DNAs. Okay, tiny DNAs. And then okay, what's the difference between gene and DNA? Gene is a sequence of DNA. Okay, it's a section of DNA. Okay, and then DNA, if you zoom in, they are blocks. So each block, we call it nucleotide. This is the basic unit of DNA. Okay, so can you imagine a cell is already so small, nucleotide is even smaller. Okay, so very, very, very small. So this is a diagram for you. So you have chromosome, you have so this is DNA. Okay, so this is DNA. Can you see DNA? And then a sequence. Let's say there's a sequence here. That's what we call a gene. Maybe this is a height gene, eye color gene, and whatnot. Okay, and then let's look at our nucleotide. Okay, your nucleotide looks like this. Okay, if you really want to study this subchapter, you only need to study this part, which is to label the nucleotide. That's all. Okay, the rest of it later, it's all K-Butt and out of syllabus stuff. Okay, so we'll talk about that later. Okay, a nucleotide looks like this. Okay, so you have a pentagon here. Okay, a pentagon here. So this pentagon, it's called a pentose sugar. Okay, a pentose sugar. But in your case, you say deoxyribose sugar because deoxyribose is the, I would say, scientific name. Okay, pentose sugar is mainly for those who study just science, not biology. Okay, and then you have a phosphate group, like I told you last week. When you say 4, your mouth is rounded, so phosphate group is the round one. Okay, and then you have the nitrogenous base. This is our concern here. Normally in exam, they'll put a square there lah. Okay, so a nitrogenous base. Okay, you have 4 types of bases, which is GACT. Okay, GACT. Okay, 4 bases. These 4 forms the password of your cell. Okay, so can you see here? Let's take for example. Can you see ACTGCA? Okay, and then you have a sequence going on. How long is your DNA? Millions. So can you imagine millions of bases here? So this is why we are different. We have millions and trillions and of nitrogenous base. Of course, there is difference. Even between a pair of twins, somewhere along the line, along this DNA, there will be some differences. Okay, yeah, they are very similar, but there still are differences. Even your siblings or whatever. Okay, unless you're a clone, then different. Clone means I do the exact copy of your 46 chromosome. Okay, but we have not cloned a human yet. That would be a very wrong thing to do. Okay, that would be a very unethical thing to do. Okay, so I digress. Okay, let's not talk about that. So your base has GACT. Okay, G will always pair with C. Okay, so A will always pair with T. So can you see here? If you are A, you pair with T. C, you pair with G. T, A, G, C, C, G, A, T. So will they go wrong? No. Okay, it's like a lock and key shape. They only fit each other. Okay, so this sequence, everyone is different. Okay, everyone. There's no 2 individuals that has the same DNA. practically impossible. Okay, so this is how it looks like. Okay, this is basically your nucleotide lah. Okay, so this whole thing we call it DNA, which stands for deoxyribose nucleic acid. Okay, deoxyribose comes from the sugar. Okay, deoxyribose, and then it's called nucleotide. That's why I call it nucleic acid. So it's called DNA. Okay, that's the short name of it. Okay, so made out of nucleotides. Okay, we talked about this already. So what's a nucleotide? It consists of a pentose sugar, okay, called deoxyribose sugar. Okay, and then you have a nitrogenous base. Okay, one or more phosphate. This one you don't need to know one or more. Okay, you just remember it's one phosphate group. That's all. Okay, so how a trait is manifested from the basic unit of inheritance. So how did your dad or mom pass something to you? Okay, so you have, let's say, gene 1, okay, gene 2 and gene 3, okay, sequence. Let's say this is height gene, this is eye color gene, this is your eyelid, single eyelid gene or double eyelid gene, whatever. Okay, so chromosome which contains DNA present in the nucleus. Okay, so a specific sequence of nitrogenous base in DNA is called a gene. Okay, we know this. A gene codes or stores message that determines how a specific polypeptide chain should be made in a cell. So this gene is your basic setting. Let's say your gene is a tall gene, your basic setting is tall, you will most likely be tall. You will easily be tall. Unless you don't get enough nutrient, then that's a different story. But most likely you'll be tall. Okay, so this is what we call transcription. Okay, do you still remember? A will pair with T, C will pair with G. So A pair with A, what is this U? Okay, in RNA, okay, what is RNA? RNA is the messenger that carries your DNA information to the ribosome. What's the function of ribosome? Ribosome is to make protein. You live to make protein. Your hair is protein, you make hair. Your nail is protein, you make nails. Okay? Your enzyme is protein, you make enzyme. Your hemoglobin is protein, you make hemoglobin. Basically, your whole body makes protein. Okay? So, all these instructions, okay, imagine building a protein is like building a machine. So when you build machine or you buy, let's say you buy a furniture from IKEA, you can't just put them up. Okay, nobody is that smart, not me anyway. You need to look at the manual. There's a manual there. Okay, if without manual, I don't think you can make anything. So same thing here. The manual is the DNA. The manual is the DNA. So anything, they will refer to the DNA. Okay, they will refer to the DNA, and then the ribosome can't go into the nucleus and read the DNA directly, so they need a messenger. That's why we call it the mRNA. M stands for messenger RNA. Okay, you have two RNA actually. You have tRNA and mRNA. Okay, if this is too confusing for you, doesn't matter. Very seldomly asked in exam. Okay, but if you are very interested, okay, you can go on and learn. Okay, so can you see A pairs with G? Okay, U. Sorry, U is the same as T. Okay, in RNA there's no T. It's substituted by a U. Okay, so T is the same as U. So can you see A pairs with U? Okay, C, G, C, G, A, U, A, U, A, U, C, G, C, G, G, C, A, U, G, C, T, A. Okay, they are, you won't get G, A, impossible. Okay, so this is the message. This is the original message. This is the photostated message. Okay, so the photostated message will go to your ribosome, and then your ribosome will start reading from this instruction. This is like the instruction, okay, like a military instruction. Okay, if you watch military film, you know, oh, when the telegram comes means there's a order from the headquarters, you're supposed to bomb the enemy or something. Okay, so let's say UGG, okay, this is what we call a codon. Okay, every amino acid has a code. Okay, let's say UGG is, I think TRP is trypsin. Okay, I couldn't remember. PHE is phenylalanine. Okay, phenylalanine. This is a type of amino acid. And then GTY, I think is, I couldn't remember. Okay, there are 20 of it. Okay, so each one has a code. Okay, so let's say UGG is trypsin, UUU is phenylalanine, GGC is whatever. So they will just put, remember, amino acids like the Lego, so I will put the Lego together according to this. Okay, so what happens if you have a genetic disease? Okay, if you have a genetic disease, maybe this thing becomes a U, becomes a C. Okay, and then this thing will become a G. Okay, So if this is UGU, it's not phenylalanine anymore. It's another amino acid. Let's say this is glutamine. Okay, glutamine is a type of amino acid. So when you change one amino acid, you change the primary structure. Remember your form 4? You change your primary structure. Your primary structure changes, you change your secondary structure. You change your tertiary structure, you change your quaternary structure. Your whole protein will look absolutely different. This is why mutation is such a serious thing. One change in a base can change everything. It's like a chain reaction. That's why people have anemia, thalassemia, sickle cell anemia. Sickle cell anemia is when one base change, it changes the mRNA, it changes the protein, which is the primary structure, it changes the secondary, tertiary, quaternary, the whole shape of the hemoglobin changes, it becomes a sickle shape. It's not round anymore, so you can't carry oxygen. Such a small error can lead to a huge error. This is why. Okay, this is why mutation is such a... And what is it so serious? Because it's so small, we cannot see. It's not like there's a change in a base, you know. No, you don't know. Okay, it's such a small thing, you won't know until you see the effect manifested in your body. That's why it's very hard to tackle genetic disease. That's why you don't know which base went wrong. Okay, hope you understand this. Okay, so mRNA, so it goes to a protein, and then your protein will be secreted by your living tissue. Okay, so your body does this every day. Okay, if your body want to make hair, let's say you cut your hair, your body wants to grow hair, so they will refer to the hair gene. So they will code the hair gene. Okay, and then maybe your hair is straight, or maybe your hair is curly. It depends on the protein inside. My hair is straight hair. If your hair is curly hair, you have a different protein structure than me. That's why your hair is curly, my hair is straight. Okay, hair is made from protein, yeah. I hope you know this. Okay, so different. We are different. That's why we make different protein. Okay, we refer to our instruction manual, which is our DNA. Okay, so I hope you are getting this. Okay, so where can we apply this in real life? Okay, I wish to emphasize again, from this part onward, it's purely all K-but stuff. Okay, so they might be asked in essay. I wouldn't recommend you focus too much. You just need to know the big picture. Okay, so first thing, DNA fingerprinting. Okay, so DNA fingerprinting. You know what's fingerprinting? Okay, fingerprinting is we need to investigate something, crime scene or whatever. Okay, if they find that your fingerprint in a crime scene, then maybe you are there. Okay, so DNA fingerprinting is very much the same. Only this time we are using your DNA. Like what? When you are at a crime scene, for sure you will leave behind some hair or your skin, or maybe if a rapist will leave behind semen, sperm. So any protein can be traced back to you, because that protein only you have it. Like I said, everyone has that different sequence. Okay, so DNA fingerprinting, how does it work? So they will put an enzyme DNA to cut your DNA into pieces. Okay, to cut it into pieces, and then they will use actually this is how it works lah. So you have a pool here, you have all these fragments here, so they'll put electricity here, they put it in water, so you have positive and negative here. Roughly it works like this lah, it's not exactly like this, but roughly it's like this. So the DNA will be pulled by the electricity to here. Okay, so all these fragments will form lines on the water. So this line is the fingerprint. Okay, so if let's say on the crime scene, this is the DNA sample found, this is the DNA fingerprinting found. So just by doing this, you would know which suspect is there, was there. Okay, so if your DNA is found on your body's victim or victim's body, you better have an alibi, okay, to prove that you were not there when the murder or whatever happens, or rape happens. So this is why DNA fingerprinting is one of the most advanced technique to trace crimes. Okay, so that's why during the early 90s, if you look at countries like Korea or Japan, there are a lot of unsolved cases. Okay, you have rape or serial murder cases that till today is not caught. Why? Because during that time, DNA fingerprinting is not mature, nobody uses it. Now it's different story. They will 99% catch you even if there's just one murder. That's why you don't see a lot of serial killing going around today. Okay, because they will be managed. They will manage to catch whoever it is before anything further happens. All right. So how does it work? Okay. So you don't really need to know how does this work, okay, but you just need to know what's the importance, okay, to identify individual, okay, to screen genetic disorder. Okay, you can use this to identify if this person has some genetic disease, okay. So track genes responsible to causing certain disease, okay, same thing, okay. Test compatibility of potential organ donors with patients. So if your organ's protein matches with the patient's protein, then yeah, he would very likely be safe with the organ transplant. You cannot just transplant organ like that. It needs to be compatible. Okay, if you get an incompatible organ, your body will reject it. Okay, and your body's white blood cell will start attacking your organ. Okay, this is where serious stuff can happen. You can die. Okay, so advantage of using DNA fingerprinting, okay, more accurate, more information, only small quantity required. Just a piece of skin would do. Okay, so sample mixed DNA can be used. Okay, DNA sample lasts longer than fingerprints. Okay, fingerprint, it's not very easy to see, but this is super easy to see. That's why people use DNA fingerprinting. Okay, nobody use fingerprints anymore, very seldom lah. Okay, and then next, human genome project. Okay, this is a very ambitious project. They started in the 90s, I think in the 90s. Okay, they want to put all our database. Okay, just imagine you have a sequence of DNA. You are a database, so they try to put every database into this human genome project, so that next time, maybe if you have Down syndrome, a person has Down syndrome, they can use this human genome project and resequence their DNA. Is it possible today? No. This is future stuff. Maybe next time we will find a cure. I don't know. Okay, so solving genetic disease is like solving a very big Rubik's Cube. Okay, it takes a lot of time. Okay, that's why even you see COVID-19, it's almost the same thing. COVID-19, they are looking for vaccine. To look for vaccine, you need to crack the genetic code of the COVID virus itself. It takes time. Okay, it takes time. You might think, okay, how long to develop a vaccine for now? According to the news, they say it's 1 to 2 years. You may say, so why is it so long? To develop a vaccine, it used to take 10 years. Think about that. Now it's 1 year. It's 10 times faster. We cannot ask for more. Okay, we are doing the race against time to find vaccine. Okay, and it's not very easy. Like I said, it's like cracking a super big Rubik's Cube. Okay, can you imagine a 3 times 3 Rubik's Cube everyone can do? This is like a 10 times 10 Rubik's Cube. Okay, it takes time. It's super long. Okay, it's even more complicated than a Rubik's Cube. I'm just using an analogy so that you can understand. Okay, so importance of human genome to identify the names and location of all human genes. Okay, all the 22 pairs of autosome. Location, where is it? Let's say your height gene, where is it? Your let's say if you're Down syndrome, where you're affected. Okay, and then autosome X and Y chromosome, determine the nucleotide sequence. Okay, we try to sequence everything inside. Okay, it's like a database there. To identify the gene that cause human disease, okay, for example, Huntington disease, cystic fibrosis. Okay, Huntington disease is a very rare disease. Okay, what is that? You can go and Google it yourself. Okay, to manufacture unknown human proteins which have potential medical value. Okay, so this human genome project has a very huge potential. Okay, but then again, it is not very well funded. Okay, to research anything scientific, you need funds, you need money. Okay, like NASA, okay, to go to space, they need a lot of money. Okay, but unfortunately, a lot of governments, they don't really sponsor to these kind of projects. Okay, that's why they are relying on billionaires like Bill Gates to donate to them so that they can carry on this project. Okay, a lot of governments, they simply didn't care. They don't care. Okay, like the US government, they don't care. They would rather spend money on military. Okay, that's a sad thing to see. Okay, we are like 2020, we are still having guns, having rockets, having missiles to fight among ourselves, but we are not funding this type of things to cure genetic disease. Okay, pretty sad if you ask me. Okay, we don't care about each other's well-being, we just care about killing each other more. Okay, so anyway, stem cell research. Okay, this is also a very new technology, very amazing technology. So special cell with unique ability to reproduce itself over a long period. Okay, what is stem cell? Stem cell is undifferentiated cell. Okay, do you still remember you will do cell differentiation? Okay, you start from embryo, and then your embryo will do cell differentiation to become follicle cell, to become muscle cell, to become nerve cell, and whatever. So stem cell is the undifferentiated one. So where is it located? You have stem cell, but the most stem cell you have was in your umbilical cord. Okay, that's why some people, they store umbilical cord when they are born, so that, is it going to do anything now? No, it's for future. Maybe next time we can use this stem cell to grow another organ and then to replace your organ when you're old. Very ambitious. Is it possible? Not yet. They managed to clone a small patch of skin, okay, and use it to cure diabetic patient. To grow an organ, we are still very far away. Okay, but then you might ask, so why are they stupid? Why are they storing it? This is for future. What if one day they found a cure? Then you say, oh shit, I don't have my stem cell. Then gone case lah. Okay, you're not going to do anything. So embryonic stem cell, okay, the cell derived from blastocyst or the umbilical cord. Okay, this is what we store. This cell can be differentiated easily into all types of specific tissue. Can you imagine you can grow muscle cell out of you can put it in petri dish, and then you grow muscle, and then maybe you replace another torn muscle. That is freaking amazing if you ask me. Okay, maybe you can cure nerve cell. Okay, nerve cell cannot do mitosis. When you break your nerve, let's say you break your spine, you are bedridden, you are not going to walk for the rest of your life. What if next time we can use stem cell and grow nerve cell and replace your spine? Very ambitious, okay, it's very difficult, but it's not impossible. Who knows, one day it will happen. Okay, and then adult stem cell, you have stem cell now, just very little. Okay, so obtained from many organs such as brain, bone marrow, skeletal, and blah blah blah. Okay, can be differentiated into specific tissue. Okay, so the stem cell we store is usually this. Okay, so what is the importance to replace disease cell? Okay, can differentiate different types of cell. Okay, it might be a future. Okay, that's why you see it's not much information here, because it's not a very mature technology yet. Okay, so genetic engineering, okay, we use this all the time. Okay, now we are developing the COVID vaccine, like I said just now, it's all genetic engineering. Okay, so genetic engineering is very useful. So a technique of gene manipulation to create new combination of gene involve transfer of DNA from one living organism to another. Okay, so where do we use this? Okay, one of the example is to produce drugs and vaccines. Okay, so let's talk about, yes, insulin. Okay, very good example here. Okay, last time, if you are diabetic, last time I'm not talking about hundreds of years ago, I'm talking about just maybe 20-30 years ago. You don't get insulin, not from humans anyway. Okay, if you are diabetic, you need insulin injection. Tough luck, they can only get insulin from bovine, or bovine means cow lah. Okay, or pig. Okay, they can only get it from animal source. Okay, which poses a huge problem because some people, they are vegetarian. Okay, they are vegan. And second, the bigger problem is it might not be compatible towards human. Okay, because you put an animal stuff into human, of course there's going to be a big chance of rejection. So what they did, okay, in genetic engineering was, okay, they take a healthy human cell, okay, and then they find the gene responsible for making insulin. They extract the thing out, okay, and then they forcefully insert into the DNA of a bacteria. Why a bacteria? Because the bacteria can reproduce very quickly. Okay, not like us human, we don't really do mitosis that much. So when you insert this, so the bacteria will start duplicating, okay, and then it will start following. Like I said, DNA is the instruction manual. So whatever instruction manual you insert into the bacteria will just read and follow the instruction. So it will produce a lot of insulin. And what is good about this insulin, it is all from a healthy human gene. It's 100% compatible to human insulin. Okay, that's why nowadays insulin has become so cheap. Not very cheap, but cheaper than last time. It's become so cheap. It's being mass produced. We solve a lot of diabetes problem because of this. Okay, this is what we call a recombinant technique. Okay, we put whatever DNA into a bacteria and let them grow. Okay, this is how we produce drugs. Let's say you want to produce paracetamol, you use this method. Any vaccines, to mass produce vaccine, this is the method you use. Okay, it solves a lot of problems. All the drugs you buy from pharmacies is produced using this way. Okay, this is why this is such a groundbreaking way to. Okay, so that's why medicine nowadays is so cheap. Okay, you go to pharmacy, any drugs you apply on your skin or painkiller, it's so cheap nowadays because it's mass produced using this way. Okay, pretty amazing technology if you ask me. Okay, so in agricultural field, okay, this one they will ask in essay. So what do? They use this to produce GMO, which is genetically modified organism. Okay, maybe this strain of paddy. Okay, maybe this paddy I modify a little bit, I can increase 20% the yield per year, or I can increase the vitamin content in this paddy. Why not? Okay, why not? People can eat less food and get more vitamin. Okay, so this is what they mean by GMO. So there are some people who say that, oh sir, GMO, I heard it can cause cancer. No, it's not proven. It is not proven. Okay, please don't buy into all these stupid, oh, GM is bad for you. Okay, the sun is bad for you. Do you put sunscreen? Sun is a confirmed way that you will get cancer. You will get skin cancer. Do you put sunscreen? No. None of us put sunscreen. When's the last time you see people put sunscreen on their skin? No. So don't worry about this GM thing. You're not gonna die. Just eat. Okay. So you acquire one more gene by artificial means. Okay, if the gene is from another species, it becomes a transgenic organism. Okay, I can even produce a better crop. Okay, if I can increase the crop by 20%, I solve more world hunger. Why not? Okay, I don't see any bad points in it. So what's the importance? Okay, to increase quantity and quality of meat. Maybe the meat I can make it leaner, less fat, so that I can reduce heart problem. Hey, that's a win. Okay, and then increase milk yield, okay, for cows, okay, to produce GM food, okay, like tomato, maize, soya bean, okay, I can make them more nutritious, okay, produce plants that are resistant to disease. It's very important. Maybe I change a little genetic of this plant, they can withstand a lot of disease and weather. This thing can last longer. Last longer means more food means less hunger. Okay, world hunger is the thing that we need to solve. It's still not solved, people are still hungry. Okay, obesity rate is higher than world hunger rate, which is ridiculous. It means we have a lot of food, but we just don't share it around. Okay, so I think that's it for this week. Okay, so next lesson we'll go into variation, which is the last part of this chapter, and we will be pretty much done with the syllabus of Form 5. Okay, so quick, isn't it? You are having SPM next year, so you have 6 months to prepare. Okay, so next let's look at our exercise. Okay, so this will be a very short exercise. Okay, so nucleotide is a basic unit of DNA, shows the structure of a nucleotide, name structure X and Y. Okay, so X is my deoxyribose sugar. Okay, Y is the square shaped thing, so that's my nitrogenous base. Okay, so this is a nucleotide chain. Okay, you draw. Okay, they want you to draw another one. Okay, I didn't show you how to draw just now. Okay, so basically it's the same thing. Okay, whatever shape this is in, you pair it with another, because you remember G, C, and A, T, they pair with each other. Okay, and then when you draw this, please don't draw it like this. Okay, that's wrong. You need to draw it flip upside down like a mirror. Okay, so instead it looks like this. Sorry, it looks like this. Okay, let me finish this and you'll understand. Okay, yeah, it looks like this. Hang on, it looks like this. Okay, so can you see it's a reverse? The sugar is reversed. Okay, so when they coil together, that's why you get a double helix shape of the DNA. Okay, so I didn't show you this just now. Okay, and oh, there you get 3 marks there, so easy. Okay, so character of human. Oh, this is next week's stuff. Okay, let's not go into this. Okay, this is last week's stuff. Okay, I hope you know how to do this. Okay, homozygous brown fur. Okay, imagine this is big B, big B. Okay, homozygous white fur, small b, small b. Okay, oh, you get all white, sorry, then white must be the dominant. Okay, so big B, big B. Okay, or we don't use B lah, because this is white, so we use W. Whatever lah, same thing. So W, W, this is W, W. Okay, so when you pair them, you get W, 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 W. That's why you all get white fur. Okay, So why are all of them have white fur? Because white is the dominant gene, okay, and they are, what do you call this? We call it heterozygous, okay. So let's label this again, okay. This is looking a bit weird, but you know, it's the same thing, okay. So label all the parts, okay. So A pairs with T, okay. C pairs with G, okay. So label the parts P and Q, okay. So P, since this is this thing, this is your deoxyribose sugar, okay. Q is your nitro, wait, this is a very weird diagram, okay. Q is a phosphate group. I think they use the wrong shape lah, okay, because your nitrogenous base is supposed to be square, which means this thing should be square, this thing should be round, okay. Tricky question. Okay, so I use appropriate letter to indicate the identities of unlabeled bases on the diagram. Okay, so just write everything. So T is A, G is C, just like this. Okay, how are the pairs of nitrogenous base in the two chains held together? Okay, so how are they held together between here? Okay, we use something called hydrogen bond. Not important, this is college stuff. Okay, draw a line to show where one pair is held by the bond. Okay, so you can just draw, okay, C and G, they have 3 bonds in between. Okay, A and T, they have 2. Why 2? Why 3? Don't ask me, this is college stuff. Not gonna tell you. Okay, what is the molecule subunit labeled S? Okay, so this S, one whole unit, we call it nucleotide. This one, it's your syllabus. Okay, the rest is not. Okay, in an investigation, the police discovered blood stain in the crime scene. They took samples, okay, conduct DNA fingerprinting. Okay, 3 suspects to be believed to be involved. Okay, so who matches? Okay, what is DNA fingerprinting? Okay, you know, I'm not going to explain this. Okay, write it yourself. Explain why DNA fingerprinting can be used to identify a person. Okay, you know what? Let's skip this. Not really important. Let's get question 3. This is the recombinant one. Okay, a gene of interest can be removed from I05 from a chromosome of donor DNA and cloned in a bacterium. Okay, this is like my insulin thing. Okay, explain the benefit and risk of application of genetic engineering in the medicine and agriculture. Okay, so you have 5 marks here and then 5 marks here. Okay, so you can give 3 for medicine. Okay, for medicine, you can use it to manufacture, okay, insulin, okay, identical to human insulin, okay, lower chance of rejection. Okay, how about agriculture? Okay, to produce strains resistant to diseases. Okay, to produce higher yield. Okay, risk, there are so many, okay, you can write it yourself. Okay, not going to go into that. Okay, so DNA fingerprinting, not going to go into this. Okay, not much here. Okay, so I think I will stop here. Very short lesson. If you don't know how to do all this, no worries. Normally they will be asked in essay question, which you can choose not to do it. I wouldn't recommend you to do this because it might be go out of syllabus. Okay, so next week we're going to variation. Again, I still wish to stress that the important part of this chapter is the matching one, the 1,3,1,4,2,3,2,4, Mendel's first second law, that one. And the blood group we learned last week, okay, all the sex-linked disease, color blindness and hemophilia, what is carrier female, that is the important one. This one, not so much, wouldn't focus. Okay, so next week we will go into the last but the most important part, one of the most important part of chapter 6, okay, which is variation. Okay, so that's it for this week. Okay, give me a thumbs up if you find this helpful. Subscribe to my channel. if you are interested in joining our live class, we are opening our class on the 15th of July. Okay, you can choose to come to our physical class or you can come attend our live class. Okay, so if you're interested, kindly go to TDC Education on Facebook or you can search it on Google. All right, I will see you again next week. Till then, and bye bye.